So, good afternoon, students. Good afternoon, students. Today we will go for the next topic, that is the plant breeding for disease resistance. Plant breeding for disease resistance. Here in this topic, we will see one term, that is the pathogen. What is the pathogen? Here in the plants, there will be some specific agents. What it will be? There will be specific agents which causes the diseases. Here, agents are the organisms which causes the diseases. So, what are the pathogens or the organisms or the agents which is going to cause the diseases? So, there are several or wide range of pathogens are there in the environment which is mainly causing the diseases to the plants. What are that particularly pathogens in which is causing diseases in the plants? Means that is the fungi or fungal diseases, the bacterial diseases, which is caused by the bacteria, okay, and one more, that is the viral diseases. These are some of, according to the pathogens, these are some of the diseases which can be seen in the crops. So it is not only affecting to the crops, okay, at some extent it will destroy or damage the 20 to 30% of the crop yield. Okay, it will damage or it is going to affect. How it is going to affect means by damaging. Okay, damaging of around 20 to 30% of the crop yield is going to damage sometimes. But sometimes what happens means total crop yield is going to destroy or it affects to the total crop yield, by which the farmers or whatever the crop yield will be there, they are going to, there will be in the totally loss. Why? Due to the causing of some of these fungal, bacterial or viral diseases. In this situation, they had practiced a process that is known as the breeding. Okay. In this situation, whatever the situation of causing the diseases and due to which there is a loss of crop yield or crops means at that time they had developed, to overcome from this problem, they have developed a breeding procedure. Okay, plant breeding procedure, and they had developed, by breeding procedure, they had developed a cultivars. Cultivars means new varieties they had developed. For what? And that cultivars will be of disease resistance. What they had developed by implementing the plant breeding plant breeding technique means that is the production of cultivars. Cultivars means some of the new plant varieties with the disease resistance in different crop yields or in different species of the crop yields. Okay, so which will this process of developing the breeding process by which they are developing the cultivars, due to this all over there is an, and in this way they are enhancing the food production. What they are doing? They are enhancing the food production. Okay, in this way they can enhance by producing the some of the disease resistance plant by what? By plant breeding technique. So let's see. See, by using this technique also they may reduce the usage of what? Of bactericide. Bactericides means some of the chemical substances which are used for killing the bacteria as well as the fungicides. Fungicides means these are some of the chemical substances which are used to kill the fungal infection or fungi which is going to cause the disease fungal infection in the plants, as well as there will be certain chemical substances which will kill the bacteria. So to them we are calling it as the bactericide and fungicide, but it has a wrong impact on the whatever the crop. Okay, it has a very disadvantage, or it has a harmful effect on the human body. That's why it is by using this technique, that is a plant breeding technique, they are developing the certain what we call a disease resistance plant, by which they are reducing the usage of what bactericide and fungicide. So next here, let's see how they are reducing the resistance host plant or resistant plants to the diseases. Here you see that is a resistance to host plant. Resistance to host plant. Whatever the plant in which there is a presence of some disease resistance genes or disease resistant pathogen, or whatever the see, if a plant is there which is having a ability of disease resistance, to them we are calling it as the host plant. So here that is resistance to host plant. So this host plant has the ability to prevent pathogens. Whatever the host plant which is improved, which is developed for what? For disease resistance, that particular host plant will have the ability to prevent the pathogen. So that that particular plant, near that particular plant, there will be no growth of fungi, or there will be no surviving of bacteria and causing the infection or diseases in that particular plant. So that is it has the ability to prevent the pathogens. Okay, the next thing here, so pathogens from causing the diseases. So this is how this host plant is identified or determined, that is by the genetic constitution. This host plant is determined by genetic constituents. See here what I have to explain means. So this is the one plant. Okay, this is one plant to which we are calling it as the host plant. What we are calling? We are calling it as the host plant. Okay, this host plant, whatever the host plant is there, that host plant has one ability. What? Near to this host plant, see, for example, this is the fungi, which is going to cause the diseases. This is the fungi, and some of the bacteria will be there. These are the bacteria which are present in the soil, especially in the soil. These are what? These all are pathogens. So here in this host plant, there will be presence of certain genes. These are the genes. So how we can come to know that particular plant is a disease resistance, or particular host plant is a disease resistance means by knowing their genetic constituents, by knowing their genes, their genetic characters, we can identify that particular host plant is preventing the pathogens or preventing the diseases. Okay, so here they have discussed some of the fungal diseases in the plants. Let's see here. See what it is means some of the disease causing fungi. Disease causing fungi in the plants. And what are that? See, I told you there are certain specific agents or the pathogens which are going to cause the diseases in the plants. In them, the whatever the one thing is that is the fungi. There are some fungi which is going to cause the fungal infection or the fungal diseases in the plants. And what are that fungi means? Here they had given, that is rust. Rust, it's a name of fungi which is going to cause the disease in what means, especially in the wheat. What is that disease name? It means that is brown rust of wheat. Okay, there will be certain rust. Some fungi will be there in wheat, which will be of brown color. That is the brown rust of wheat, which is going to cause the diseases in the wheat crop fields. Okay, the next red rot of sugarcane. Have you eaten the sugarcane? Sometimes, if this is the stem of the sugarcane, means in between that particular stem, you can find some red color spots. Okay, here in between that one, in the middle region of the sugarcane, there will be presence of some red spots. What is that red spots means? That is also a fungi which is grown, which is surviving in the sugarcane, and that is causing a disease. And the name of that disease is the red rot of sugarcane. The wheat, which is the disease, that is the brown rust of wheat. The next here, one more disease is there, which is going to cause one more fungi is there, which is going to cause the disease in the potato. And the name of that particular disease is the late blight of potato. Okay, here on the potato some spots are going to cause, that is the fungal infection or the fungal disease, and the disease is known as the late blight of potato. Okay, so these are some of the fungi which is going to cause the which is going to cause the fungal diseases in the crop fields. So here there are certain examples I have told you: brown rust of wheat, which is caused by the rust; red rot of sugarcane; late blight of potato. This is about the fungi. Some of the bacteria or bacterial diseases we will discuss here. That is by bacteria, which disease is going to cause in the crop yields? That is the black rot of crucifers. In the crucifers, a bacterial infection is going to cause, and that is known as the black rot of crucifers. Okay, next that is by the viruses. Some examples are there by viruses. What is the example of that disease? That is tobacco mosaic virus, or TMV also you can say. That is tobacco mosaic virus. Then accept that one, turnip mosaic, etc. Okay, this is about the diseases which is caused by the viruses, and that is the diseases which is caused by the bacteria. By bacteria, which in whom you can see the bacterial disease means that is especially in the crucifers. Okay, that is the black rot of crucifers. The next by viruses, what it is means TMV, that is in the tobacco plant. Okay, tobacco mosaic virus, and in the turnip plants you can see a virus that is known as the turnip mosaic virus. Okay, or turnip mosaic. So this is about what some of the diseases with a specific agent or with a pathogen, that is the fungi or virus or the bacteria. The next, what are the methods for of breeding for disease resistance plants? So let's see what are that methods. Methods of breeding for disease resistance plants. Methods of breeding of breeding for disease resistance plants. Okay, what are that methods? Here there are certain methods. What are that means? That is, one is conventional method. One more, that is mutational breeding. Sorry, conventional breeding, conventional breeding method, or by mutational breeding. These two methods are there. One is conventional method or conventional breeding technique, and one more, that is mutational breeding technique. Okay, so first we will see for the conventional breeding for disease resistance plant. Here in the conventional breeding resistance plants, whatever, see, conventional resistance for disease resistance is that of hybridization and selection. Here the two processes are very important in what? In the conventional. See here, first I will explain this one, then we will go for the mutational breeding. Here in the conventional breeding for disease resistance, the two things are very important, is that hybridization and selection. Hybridization and selection. Crossing the two different plants of different species or different varieties, whatever the hybrid they got or the progeny they will get, that hybrid or progeny they are going to select. For what? For further crop yield. Okay. See here, that is the first and foremost very important thing in what? Conventional breeding method. And the next, its steps are essentially this, whatever the steps involved in this method is essentially similar to the breeding for other agronomic crop yields. Okay, whatever we had discussed in the last classes about the plant breeding technique, there are 5 steps. That steps only you can see here in what? Here in the conventional method for breeding for disease resistance plants. So this whatever the steps are essentially identical to those for breeding for any other economic characters such as the high yield or some other characters, whatever the breeding procedure is there, that procedure only you can implement on this particular method. The various sequential steps, what are the steps which are involved or which is these are the steps which are involved in the conventional breeding technique, that is the screening the germplasm, first one, screening the germplasm, for resistant sources. Screening the germplasm for resistance sources. See, germplasm means collecting the seeds or plants which have a disease resistance. Such a type of seeds or plants they are going to collect. Okay, that is the screening the germplasm for resistance sources. Then next, hybridization of selected plants. Then by collecting them, they are making them to cross. That is the hybridization of selected plants. Hybridization of selected plants. Okay, this is the second step. Then third step, selection and evaluation of hybrids. Whatever the hybrids they will get, see, first they will select whatever the germplasm, then they will make them to cross. That is the hybridization of what? Of selected parents or selected plants, selected parents. Okay, by selecting such as seeds or plants, they are making them to sexual hybridization. Okay, then next, after hybridization, whatever they will get, the progeny or the hybrid, that is going to be selected for further crossing purpose. Then lastly, the last step, testing and release of release of testing and release of new varieties. The last step, where they will test under different agronomic conditions, then they will release into the different crop fields, that is for more than 3 seasons. They will grow in the different crop fields with different climatic conditions. Okay, then they will release them. Okay, so this is about the different steps in what conventional breeding technique. Here there are some crops, varieties breed by hybridization and selection. If not, if these steps are not going to be implemented, only by 2 things or 2 processes they can breed the plants for disease resistance. What are the 2 points means, or 2 processes, that is the hybridization and selection. Directly they can cross, then directly they can select the particular plants. So they can select for the fungi, viral, bacterial diseases, okay, which are going to released. Here some examples are there. Some crop varieties hybridized for disease resistance. What are that crop varieties? What are that disease resistance? We will see here. See here, what are that one? Some crop varieties hybridized for disease resistance. Some crop varieties hybridized for disease resistance. Okay, so see here, first crop variety, and last is the disease resistance. So let's see one by one over here. First one, that is a wheat. Okay, first wheat variety is there. What is the variety name? Is Himgiri. This is the new variety which is developed by conventional method. For what? For plant resistance. Okay. And what is that resistance to disease? It's a disease resistance means leaf and strip rust. Leaf and strip rust. This is one of the fungal disease. Okay. This resistance is employed or implemented by what? Conventional breeding. Okay. Hill bund. One more is there. That is hill bund. Okay. The next second one, which is the second crop, means brassica. Brassica is the second one. What is the new variety which is developed as a disease resistance means Pusa swirling. Pusa swirling. Okay. And what is the disease resistance means that is white rust. This is also one of the fungi, white rust. Okay. Then next, the third one, that is the cauliflower. Cauliflower. See, whatever the table I am going to draw, whatever the variety, whatever the disease resistance I am explaining here, this questions will come for 1 mark, or it will be helpful for your NEET or CT exams. Okay, from this table they will ask some of the questions. So cauliflower, Pusa shubra, Pusa shubra, and one more is there, that is the Pusa snowball K1. Pusa snowball K1. This is the new variety of what? Of cauliflower. The next, what is that particular disease? Black rot and curl bleeder. Black rot and curl bleeder. Black rot and curl bleeder. And here by Pusa in this Pusa snowball, that is the black rot. Okay, the next, that is the cowpea. Cowpea, variety name is Pusa komal. Pusa komal, and what is the disease means bacterial bleeder. Bacterial bleeder. The next, that is the fifth one, that is chili. Okay, what is the variety means Pusa sadabahar. And what is the disease resistance means chili mosaic virus. Okay, one more, that is tobacco mosaic virus, MV, tobacco mosaic virus, and leaf curl. Okay, so this is about the certain crops, their new varieties, and that varieties, then whatever the disease resistance to that particular varieties. So see here, first one, that is the wheat. Here the wheat is there, and the variety which is developed or improved by the conventional method of breeding. Okay, what is the name of that variety? Himgiri. For what? For the wheat. Okay, then here you see what is that particular disease which is causing in the wheat, which will not come in the new variety of the wheat plant, means that is the leaf strip rust. on the leaf there will be some rust, brown color rust is you can see on metals and all how the rust is going to occur like that on the leaves of the wheat. There will be some rust. Okay, that rust will cause the diseases, which is one of the fungal infection. And the next hill bundle. The next come to the second point, that is the brassica. In brassica, which is a new variety, means Pusa swarnim. What it is? Pusa swarnim, and it is the white rust. That is also one of the fungal infection. In cauliflower, you can see some black spots on the flowers of the cauliflower. Okay, on the whatever the buds will be there, on that you can see some black color spots or black colored something on the surface of the cauliflower. So they had developed one new variety. The two new varieties they had developed, that is the Pusa Shubhra and Pusa Snowball K1. And what is that particular what you can see, which disease you can see in that particular cauliflower leaves, that is the black rot and curl bleed. I told you black colored spots you can see and curl bleed. Then in Pusa Snowball also is meant for or improved or breeded for what? Black rot. Then next that is the cowpea. In cowpea, what is the new variety means, that is the Pusa Komal, and what is the disease resistant means, that is the bacterial bleed. The last thing, that is in the chili, Pusa Sadabahar. Then what is the viral infection is going to cause in the chili plants? What are the diseases? Chili mosaic virus, tobacco mosaic virus, and leaf curl. In the viruses are going to occur, except that one, whatever the plants of the chili will be there, their leaves are going to curled. Okay, so this is about some of the what we call some of the diseases, varieties, and the crops by what? By the conventional method they had developed. See, conventional breeding is often constrained by the availability of limited number of disease-resistant genes. Okay, if in the host plant there is a limited number of genes, that genes which are responsible for what? For the disease resistance that are present and identified in various crop varieties or wild relatives. First they had identified. Okay, and second thing, inducing mutations. Next comes the second. We will study in detail about the mutation breeding in the next phase. So before going to that, in conventional method also, by if there will be inducing mutation means some mutants will be there, such as some chemicals will be there, some radiations will be there, which is going to be implemented on that particular genes, by which they will change the base sequence of the genes. Okay, due to which there will be change in the genetic characters of what? Of the plants. So here, whatever inducing mutation in plants through diverse means, and then screening the plant material for resistance, sometimes they will give a desirable characters. They are inducing the mutations in the plants for what purpose? For disease resistance. But sometimes what happens while they are taking for screening, they will identify some of the desirable characters in that particular plant. Okay, and plants those have their desirable characters can then be either multiplied directly or can be used in the breeding process. So other breeding methods which are used are selection among our soma clonal variants and genetic engineering. Soma clonal variants and genetic engineering. These are the other methods by which we can select the particular plant or other breeding methods except the conventional and mutation breeding. These are some other methods by which you can select the desirable plants with disease resistance. Okay, so this is about the conventional method, which is similar to the plant breeding technique, which I had explained in the last classes. The steps will be similar. Okay, by implementing that 5 steps also you can breed, but except that one, you can breed the plants for disease resistance only by one method, that is the hybridization and what? That is the selection. Okay, so this is about the conventional method. Next is the mutation breeding. Mutation means what? Means it is a process by which genetic variations are going to occur. Genetic variation means there will be some changes in the genes. Okay, so some changes in the genes of chromosomes is going to take place where in the mutation process. So how they are going to create it? Means they are going to create it through the genes or base sequence in the genes. There will be changes in the base sequence. What do you mean by base sequence? Means that is the adenosine, guanine, nucleotides and nucleosides. What you had studied in the molecular basis. So that particular base sequence, whatever the arrangement will be there, that base sequence is going to changed by what? By which process? That is by the mutation. So due to which here, if there is a change in the base sequence within the genes, which will result in a creation of new characters, and which is not found in a parental trait, which is found in a new offspring or a new progeny. Okay, so this is about the mutation breeding. So accept this mutation breeding in detail. I will explain in the next class what are the mutants, what are the agents which are inducing the mutations, that one, and what are the examples for this mutation breeding. Okay, what are the method. The next we will go for the mutation breeding in the next class. Okay, thank you. If you have any doubt, you can ask in the chat box. So here, what is the important questions over in this particular topic means explain the steps, different steps for plant breeding technique, that will be asked for 5 marks. Then accept that one. They will ask for 1 marks about these diseases, varieties. Okay. Then accept that one. They will ask for 2 marks, that is apiculture, dairy farm management, then dairy farm management and poultry management. We will ask that also. Okay, so these are some of the important questions from till what I had explained. Okay, tomorrow I will continue with the next class.